our next speaker, we're really changing gears here, so just to wrap your head around this. Corey Plate, a Thomas W. Smith Fellow and a student at the College of Staten Island, entered CUNYBA in the fall of 2019 because of his interest in creating a multidisciplinary degree in physics, neuroscience, and psychology that will be the foundation for a career as a physician and a researcher. He is the editor-in-chief of Contemporary Psychology, which is an undergraduate publication at CSI under the mentorship of Dr. Leora Yetnikov. Since the summer of 2017, he has volunteered in laboratory studies with Dr. Dan McCloskey, who's at CSI, working on a project that is the subject of today's presentation about East African naked mole rats. I have to confess that I've become kind of obsessed by them. Also known, after I Googled them, as sand puppies, which makes them sound actually more lovable to be sure. They are actually remarkable creatures, as you will learn, because they are resistant to cancer, can survive in an atmosphere up to 80% carbon dioxide, and may live up to 30 or more years. Corey's interest, however, is in a novel glucose transporter phenomenon that may well have implications for human diabetes research. Please welcome Corey Plate.
Hi, everyone. Thank you, Dr. Hartswick. I'm really excited about today's talk. It's a subject that I'm very passionate about. And the star of today's talk is really these African naked mole rats. Despite the unassuming name, they are one of Earth's strangest and most interesting animals. And today I'm going to talk to you about why I believe that's true. And then I will tell you why I think that they could prove to be a powerful new model for diabetes. Next slide. So what makes naked mole rats so special? Well, to begin with, naked mole rats are a highly social animal. They live in groups of up to hundreds of animals living across generations together in the tight quarters of an underground colony. So you can see an example of them nesting together in the image on this slide. This has taken place in a laboratory, but it's very similar to their natural environment. Now, this exposes them to extremely high levels of carbon dioxide, in excess of 50 times the air that we breathe. Now, if we were to be in this kind of environment, we would find it intolerable, and moreover, not survivable. Now, perhaps more astonishingly, naked mole rats can survive in excess of 15 minutes without any oxygen at all. Now, we believe that similar to an antioxidant, that it's possible that this low oxygen dependence is part of why they live up to 30 years and almost never get cancer. Now, all of these signs kind of point to an unusual metabolism, which is part of why we started looking into it. And as it turns out, this is indeed the case. Naked mole rats live in what we would consider a diabetic-like state. Next slide. Sorry, next slide. Oh, there we go. So what makes this property even more interesting is that there's a special caste of naked mole rats called dispersers who are responsible for establishing new colonies, and they may not even exist in this diabetic state. Now, to explain this, we need to briefly talk about insulin. Insulin is a hormone that's present in naked mole rats and humans that allows cells to uptake sugar and use it for energy. Now, it does not appear that this insulin works as it does in humans in the average naked mole rat. However, if you compare the average naked mole rat, seen in the left image, to dispersers, seen on the right image, you can see that the dispersers are larger, paler, and often have more fat than their brothers and sisters. And unlike their brothers and sisters, they can easily tolerate the air that we breathe. Now, this suggests that they have a working form of insulin. Next slide. Now, in our work, in order to investigate this insulin difference, we compare levels of GLUT4. Now, GLUT4 is an insulin-controlled pathway for sugar into the cell. So basically, think of it like a gate, and the insulin is a key, and that allows sugar to enter the cell. Now, based on our findings seen in this graph, which showed levels of this transporter in naked mole rats, seen in blue, compared with mice, seen in red. We believe, pending further testing, that this transporter, and thus insulin, is less active in naked mole rats. And this is similar to what we see in diabetes. Now, the reason we use mice as a test model for this comparison is because mice are a kind of go-to staple in labs to compare different qualities because they have a similar enough physiology to humans and because they are easy to care for and easy to breed. Now, if we're able to establish the opposite effect in dispersers, where their GLUT4 is at a normal level, this would suggest a reversal in this diabetic-like condition. And this is important because that suggests that they're able to have a switch in this functional insulin, something that we don't see in diabetic patients. Next slide. Now, this is an exciting possibility because among the many amazing things that we can learn from naked mole rats, it suggests that they may be an excellent model for diabetic treatment. After all, it would mean that they've already found the cure. And that's it for my presentation. And then we have one more slide with a graphic that kind of shows how everything got started.
Wow, that is absolutely fascinating and a very complex graph indeed. So we're going to open it up. Thank you so much, Corey, for a fantastic presentation and for sharing your passion of the African naked mole rat with us. I've always thought they were super cute, but totally ignorant and naive in my knowledge of them. So now I will.
They are cute. They're cute in person.
So we have a few questions for you already. So the first one is, what is your specific research program?
So specific research
clarification with that one.
Sorry, one more time?
And let me know if you need any clarification.
Okay, so my specific research program, I work in an undergraduate lab with the amazing Dr. Dan McCloskey. Our entire lab focuses on research in African naked mole rats because, as you can see there, for a number of reasons, they're an interesting model just because they have all of these bizarre traits that seemingly have somehow coalesced into this one species out in Kenya. So we all focus on research in African naked mole rats, specifically in their metabolism, in their utilization of carbon dioxide, and other exciting things that have to do with their brain.
Very interesting. So another question for you, Corey. What is the next step in your research, and how have the campus closure how has it impacted your work?
So sadly, the campus closures have impacted the work. We were in the middle of running Western blots where we were comparing GLUT4 in the average naked mole rat to mice again, just to confirm our findings with the graph that you saw on the previous slide. The previous slide is an ELISA, which is used to compare different levels of protein in a species, but a Western blot is a more robust test, and it would also help us to establish the significance of this finding. Now, once we're able to do that, the next step would be to compare the average naked mole rat to dispersers. And what we think we're going to see is that the dispersers have a similar level of GLUT4 to mice, rather than to a lower concentration of it.
Oh, fascinating. I really do hope you get this research out there. It seems to have a wide-ranging impact and lots of things still unknown. So the final question is, what did you find the most interesting aspect of conducting this research? Any special little bits of knowledge that you were just like, oh my goodness, I wish other people could know about this? Anything like that you'd like to share?
Oh, I mean, the naked mole rat topic is simply bottomless. I mean, my favorite thing is just to get lost in all of the literature that's starting to crop up about this animal. I mean, I didn't really even cover the half of it in my presentation. This graph here shows a little bit of the complexity of this animal. I made this graph when I first started my research based on a paper by Park, and it incorporates several other papers that I looked into. And what was interesting is that they all just led back to this GLUT4 transporter, and that's how things got started. So my favorite thing is just getting lost in the literature of these animals and seeing how they've frankly evolved a whole different system for their mode of living.
Corey, I hope you'll continue your passion for research. It's
Thank you.
infectious. Thank you so much for a wonderful presentation and
I
for this
appreciate
amazing
it.
work.